In the sperm viability test, we differentiate the live sperm from the dead sperms. So, we use different strains like eosin or eosin necrosin strain, or we also can do hyposmotic swelling test to differentiate between the live and dead sperm. So, when you use this eosin and necrosin strain, we can see, we can observe a pale and white sperm, that is live sperms, and we can see a dark pink color, so that is a dead sperm, because the dead sperm have a broken membrane. So, the stain easily pass into the membrane, into the cell, and causes the pink or reddish color. Then we have to observe the morphology of the sperm. Generally, the sperm head is around 4 to 5 mm. Next is the midpiece, which is metabolic created for the sperm, which constitute the midpiece, which produce energy for the sperm, which constitute around 4 to 5 mm long, slender, and it is 1.5 times longer than the sperm head. And next is a tail, which is around 65 to 75 mm long, very thin, which helps the sperm to move forward. And you can see different types of sperm abnormalities like a large head, small head or pin head, double head, then elongated head, amorphous head. So, these are all head abnormalities. And regarding the midpiece abnormalities, you can see bent neck, then irregular shape of the neck, then there is a cytoplasmic droplet more than the head size is present, and also very thick cytoplasmic droplets, and also very thick midpiece, and asymmetric positioned midpiece. And regarding the tail abnormalities, you can see no tail or very short tail and broken tail, and you can also see a coiled type, that is the tail is rolled around, and also the terminal droplets, cytoplasmic droplets present at the terminal portion of the tail, and also double tail, like a head, double head, you can also see a double head. And this is a normal sperm with a oval shaped head, slender neck, and a long thin tail. In addition to this sperm motility and morphology, we have to clearly, very carefully observe whether there is any agglutination present in the semen samples. So, if you see a clump of sperms present in the semen samples, then we have to note very clearly in the semen analysis report. Generally, there are three types of agglutination we observe. One is head to head, that is head and head attached to each other, and tail to tail, and also head and tail mixed together. And also we can grade this sperm agglutination. If you see only less than 10% of sperms are clumped or agglutinated or attached together, and we can see more number of free spermatozoa, then we can give plus or grade 1. And if sperm agglutination is around 10 to 50%, more free spermatozoa, then we can give grade 2 or double plus. And if we see a large agglutination of more than 50% of spermatozoa, and we can see only few sperms moving here and there, then we can give grade 3 or 3 plus. And there is a gross agglutination where we can't see any sperms moving in this field, and all the sperms were attached or agglutinated together. So, in this condition we can give grade 4 or 4 plus. So, we have 4 grades like isolated, moderate, large and gross sperm agglutination. And the andrologist should not get confused with the agglutination, sperm agglutination and aggregation. Sperm aggregation, in that condition, the sperms are attached with the debris or dead sperm or with the round cells, and they, in the aggregation we will not observe sperms attached to another sperm and making them immotile. But in the case of agglutination, we can see the sperms attached to another sperms and forming a clump or agglutinated, and it makes the sperms immotile. It makes the sperm unable to move from here and there. So, with this we can come to conclusion that we have seen the sperm count, sperm motility, morphology, then macroscopic examination like pH, volume, viscosity, liquefaction, everything. So, after that we have to give a report according to the WHO nomenclature. If all the parameters are present in the normal range, then it is called normozoospermia. If you observe no sperm, then it is called aspermia. If you observe less than 1.4 ml, we call it as hypospermia. If you observe more than 6 ml of semen volume, we call it as hyperspermia.